gentlemen, thank you very much for the introduction. And since I am giving exactly the follow-up of the story, I'm not going into details and I'm not going to repeat the introduction. So we are still dealing with the in vivo injection, in vivo manipulation and stimulation of the chicken microbiome. Right now I'm going to focus on analysis of the chicken transcriptome and selected physiological parameters. So the experimental varying production conditions you already know, and what is thing to stress that for all traits we use exactly the same individuals. So if there is something really interesting, we can nicely correlate those results from different panel of the analysis. The experimental setup for the whole transcriptome and the physiological parameters measurement was, as you see in the slide. So after the incubation, after the injection at 12 days of embryo incubations, the chickens were hatched, and then the tissues were, several tissues were collected for the physiological parameters at up to 6 data points. So at day 1, 4, 7, 14, 21st and 42nd post-hatching, the whole transcriptome analysis using chicken Affymetrix microarray was done at day 21st. So going into results about the whole transcriptome analysis, at first we look at the immune and gut tissues. There were 3 tissues which we analyzed: the spleen, the jejunum and the cecal tonsils. On these slides I'm presenting so-called Venn diagrams, which actually look at the common and different differentially expressed genes in those 3 tissues. And as you can see, just looking by the numbers of the genes which were differentially expressed, the highest number of genes which was changed upon the symbiotic stimulation was in cecal tonsil for the symbiotic 1. The symbiotic 2 in general was less efficient in terms of changing the gene expression. Next tissue which we look at it was liver, and here the numbers are quite opposite. So the higher number of genes which was changed upon the symbiotic injection was after symbiotic 2. And as I will show you on coming slides, it's not only about the numbers of genes which were differentially expressed differs between those 2 symbiotic, but also about the specific pathways which were triggered with each of the 2 bioactive substances. So at first we look at the cecal tonsils and the symbiotic 1 effect, cecal tonsils, because that was the highest number of genes changed upon the stimulation with this bioactive substance. And as you can see, most of the pathways which were triggered are related one way to another to immune response. So it's T cell activation, B cell activation, lymphocyte proliferation, regulation on the lymphocyte differentiation. So the majority of the pathways which were triggered upon symbiotic 1 injection are related, in cecal tonsils of course, are related to the immune response. On the contrary, if we look at the liver and the genes which were gene pathways which were changed after symbiotic 2 injection, they were related mostly to metabolism, to protein metabolism, protein modification, or in general biological processes. to the result of the physiological parameters, actually we measure quite a long list, a large number of them. I'm presenting here those which are more interesting and actually give more significant results. At first we look at the ALT, so it's alanine transaminase, which is an indicator of the status of the liver. And even though the statistic is not very impressive in this case, what you see, the measurements were done in 6 data points, starting from day 1 until day 42. In white, there's indication for the control, symbiotic 1, it's in blue, symbiotic 2 in green. And what we can see, the majority of the, actually in all of the data points for the symbiotic 2, there's a nice decrease of the ALT, which indicates the good status of the liver, and in our opinion it also nicely coincides with the transcriptome results which were observed for liver after the symbiotic 2 injection. The symbiotic 1, the picture is not as clear as for symbiotic 2, but in several cases we also see the decrease of the ALT in liver. The ALT level, sorry. The next parameters we look at, there are 2 pancreatic enzymes, so the lipase and the trypsin. On the left-hand side, you see the measurements which were done in pancreas itself, and the right-hand side, panel, there are measurements which were done in duodenum, and again there were 6 data points. The control is indicated in white, symbiotic 1 in green, symbiotic 2, symbiotic 1 in blue, symbiotic 2 in green, sorry. And in pancreas, the picture is actually not very clear. There are some effects like the lipase. Is there any word pointer?
Beneath. You must switch. On the other side. Yes.
Okay, so there is some transient effect which is observed for the lipase at day 7 and day 14, where we observe the higher activity of lipase after symbiotic 2 injection, but the picture is more clear in duodenum, where for both pancreatic enzymes, so the lipase and the trypsin activity is elevated after both symbiotic treatment. Most of the data point, in some of them it's statistically significant, which is indicated with the arrows. Next parameter which we look at it was total iron-free triiodothyronine, which is the indication of the elevated metabolism. And here we see the much higher metabolism in both symbiotic groups at the middle of the rearing period, actually at day 7. And last but not least, since the previous results didn't give actually very consistent, very clear picture, we decided to look at the incretins and the incretin level. The incretins, they are the enzymes which are responsible for insulin release from the pancreas. So higher incretins like the gastric inhibitory peptide and the glucagon-like peptide, the GAP and the GLP. Higher the level, high level of those incretins means also higher level of insulin. In our case, both incretins which were measured actually were down-regulated in the symbiotic groups. And this down-regulation was very consistent, so it was down-regulation in terms of the level measured in the serum, in terms of both at the mRNA level of the gene itself and also at the receptor level. And probably this parameter might explain the constant level of the FCE, so that we didn't observe the elevated FCE, what was mentioned already by Kasia, with still maintaining good body weight. So to conclude, what we observed after the symbiotic administration and the gene expression level, both symbiotics trigger different gene expression pathways. For the S1, it was mostly immune-related pathways. S2, the metabolic pathways. At the physiological level, different physiological and biochemical parameters were changed, but the effects were not very, well, they were rather transient. It was not the clear picture which we could observe for the entire rearing period. But what we did detect, it was the strong modulation of the incretins expression. And to sum up our both presentations, we concluded that the symbiotics in OVO reduced the mortality and maintained the body weight without deterioration of the FCE. And actually two symbiotics, those two which we use in this project, might be applied in production, might be applied at the farm level. It was already mentioned by Kasia, in different conditions. So if we want to stimulate more immune side of the organism, we definitely should use the symbiotic one. If the goal is to obtain, for example, the premium product and to improve the meat quality or the body weight, then the symbiotic too should be the symbiotic of choice. And that's all from my side. Thank you both very much for your attention. And thank you.